I'm Jacinda,
And I'm Erysa.
and we're going to be presenting on messenger RNA vaccines. So we'll talk about what messenger RNA is, what messenger RNA vaccines are, the characteristics, the mechanism of action, the different types, the delivery strategies, how they're administered, and recent findings. So messenger RNAs are single-stranded RNAs that as a template for protein translation, and they transfer genetic information from DNA to protein. So mRNA vaccines are created in a lab to make proteins that trigger immune response, as well as produce antibodies that protect from getting sick or infected from a virus in the future. So what are their characteristics?
All right, so let's talk about some characteristics of the mRNA vaccine. We'll talk about both positive and negative aspects. So on the positive side, it's adaptable. So it allows for quick updates, especially as new variants emerge. It also has a fast production cycle and it's cost effective. So because there is no need to culture cells or any kind of culture mediums, the production cycle is cut down and it allows for upscaling of the production cycle. So they're able to mass produce a lot more vaccines than they were to if they were using live attenuated vaccines. Right? It also lacks viral material. So because the protein has no virulence, there is no chance of being infected with the virus after receiving this vaccine. Right? And then for negative aspects, it's easily degradable. So if the mRNA is naked, meaning that it's not encapsulated by anything or protected by something like a lipid, then it's easily degradable by a ribonuclease. It's also large in size and negatively charged, and this hinders its membrane permeability. And these last three negative aspects, they allude to the importance of delivery strategies, and we'll get into that a little bit.
So what is the mechanism of action of mRNA vaccines? So we'll talk about it a little later about the delivery strategies, but when mRNA vaccines are delivered to the cells, they enter the cells and are transported to antigen presenting cells where they are translated to targeted antigen and converted into proteins. So that targeted antigen protein is degraded by proteasomes, and then they are, they are degraded by proteasomes and turned into polypeptide epitopes on major histocompatibility complexes. So they're either bind to major histocompatibility class 1, where they induce CD8 T cellular immune response, or to major histocompatibility class 2, where they induce CD4 T cellular immune response. And so what are the different types of mRNA vaccines? So we have non-replicating, which are based on recombinant viral vectors. So they have a 3' translated region, targeted antigen sequence in a 5' translated region, and they host immune response without replicating in the host cells. And then we have some amplifying mRNA vaccines that are based on alpha viruses, and they amplify the genome response and encode mRNA by mimicking viral infection. And so this results in sustained levels of combined immune and innate immune response. And so this leads to prolonged antigen-specific humoral and cellular immune response. So how are they delivered?
All right, so now let's talk about the delivery strategies for the mRNA vaccines. First, you have lipid-based. So lipid-based particles or lipid nanoparticles are going to encapsulate the RNA, and they're going to protect it against degradation. After that, you have polymer-based. So the polymer-based delivery strategy is going to ensure functional delivery of the mRNA and also protect against degradation. Peptide-based. So when mRNA is in the presence of a specific peptide called proteomine, it's going to be well protected and it's going to have an increased transinfection rate. Then we have virus-like replicon. So virus particles are able to carry and deliver self-amplifying RNA into the cytoplasm. And this is going to, after that, they're going to self-replicate and express the necessary antigens. Then you have cation nanoemulsion. This also protects against degradation. This not only protects against degradation, but it also has low cytotoxicity. and it is able to, it has low cytotoxicity, and it has, an increased transinfection rate as well. Then you have naked mRNA vaccines. So like I mentioned earlier, when the mRNA is naked, it's not encapsulated by anything. So when you do want to use a naked mRNA vaccine, it doesn't need an additional carrier. So it just needs to be dissolved in a buffer solution and then injected directly into the patient. And then finally, we have dendritic cell-based. So we know that dendritic cells are the best antigen presenting cells. So in this case, you're going to extract dendritic cells from the patient. You're going to reprogram it using mRNA, administer it back into the patient, and that's going to elicit an immune response. All right, so now that we know a little bit about delivery strategies, let's get into the administration routes. So first we have intradermal injections. So this means that the vaccine is injected directly into the dermis region of the skin. And here the antigen presenting cells are going to internalize and process the mRNA vaccine. Then we have subcutaneous. So the vaccine is administered to the layer of the skin under the epidermis and dermis. And this allows for a larger injection volume than the intradermal one does. And it allows for less pain in the injection site for the patient. Then you have intramuscular. So the vaccine is administered directly into the muscle. The muscles have a large network of blood vessels that allow them to recirculate and recruit different immune cells. Then you have intravenous injection. So this route allows for the largest injection volume of all of these options, and it delivers the mRNA into the systemic circulation. And then finally, you have intranodal injection. So the vaccine is administered directly to the peripheral lymphoid organs, and then where the T and B cells are. And this method is not really used because it's a complicated procedure and it involves the use of ultrasound for guidance. All right, so now let's talk about some recent findings. So as of 2022, scientists are currently developing a mRNA cancer vaccine, and they're hoping to use it as a possible anti-tumor immunotherapeutic option. And with this vaccine, they hope that it's going to stimulate both humoral and cellular immunity and help increase vaccine adaptability for different patients and different diseases.
Another future of study for mRNA vaccines are for therapeutics involved and vaccines involved in liver-specific diseases. So after the emergency authorization and use of mRNA vaccines for COVID vaccines, they are trying to use the models and methods to create vaccines for liver-specific storage disorders. This concludes our presentation. Here are resources. Thank you.
Okay, questions? Hey, Gayle, question?
You guys.
I wasn't
I
invited.
don't have any questions yet.
Oh, my God.
It's so good. That's right.
Yeah, it's no clear.
How did you guys get so good at presenting?
Yeah, so my question is going to be now to the audience, not to them. Okay. No, no question? Oh, my question? Okay. We are mentioned in this afternoon in the presentation about vaccine. Things that sounds are similar. But you as expert can explain to me the difference between the RNA vaccine and the spike RNA vaccine? Or is it the same?
I think they're different.
Yeah.
Different?
Yes.
Explain. You are talking about COVID RNA messengers vaccine, yeah? Okay.
Be the spike viral proteins contain, so they're specified for that, those viral or those spikes on the virus.
Well, everyone can explain to me. No idea.
The spike protein needs to be, the way it's transferred, I guess, spike protein, it can't just be transferred directly into or delivered directly into the vaccine casing or directly to the patient. It needs to be refrigerated, it needs to be encapsulated. So if you're talking about like the mRNA by itself being like naked,
No, what I find is first the material that is going to be used in each of them. First different, most important to me, although there's different that you can explain about how it's going to be the storage and all this. But the main difference, can you go first to the first slide? It's the first one. The first of all. No, the first. That's the first. Ah, that's the first. Right. Really, messenger RNA. Okay. Who present the spike? You. So. Try to answer the question between the two.
Because viral vectors.
So you will not be attention.
yes, that's.
Repeat your question.
Repeat the question. If there's a difference between the messages RNA vaccine with the spike RNA vaccine,
Yes.
so discuss between you. Tell me what is the answer.
So like what Jacinda was saying, the spike protein, you enter like an empty, like modified vector of virus, and it inserts the RNA to produce the spike proteins. But with the mRNA, it's the antibody, it's producing the mRNA for the antigen, blocking the antigen.
No. I want to review the topic. I wanna, if something that tell me if they are equal or not. And what is the base in both? If they are the same.
Both utilize mRNA.
Both do not use the mRNA.
You're talking about lipids versus proteins?
Yeah. But mRNA.
Spike protein. But it's not the complete messenger RNA.
One is protein.
One is the spike, it's an area, it's a sequencing. Here is not a sequencing. This is the information that have the mRNA. All the information that have the RNA messenger.
So just like the whole mRNA sequence? Is that what you're talking?
They all. That's what you said. In that case, it's not the whole molecule. It's the spike. It's certain area. They cut and select that area and it's going to stimulate to produce that protein that's going to make the vaccine. But in your case, it's not the spike. Yes or no? Yes. No? Yes?
For us, it contains the whole target antigen sequence.
Okay, so, this slide. Yeah. Yeah. What they do,
So that it contains the 5' untranslated region, and the 3' untranslated region, along with the target antigen sequence. Hosted, or if it causes the immune response without replicating, the spells within the host.
it could be several different sequences, it's not only this spike. Right?
Yeah, okay.
Yeah,
Yeah, I get you,
there are several. Okay,
yes.
so I believe that this is the principal difference between the two type of vaccine. One take a little for the most important antigenic determinant, and the other one is going to take a bigger area of the RNA with the information. Yes?
Yes.
Okay, very good. Listen, that's right. With the net.